Hello, we are from Group 4 and we would like to talk about our group research project titled In Silico Design of Novel Multi-Epitope Cancer Vaccine Against Non-Small Cell Lung Cancer (NSCLC) for Indonesia population. So in this video, we'll talk about the project background and our proposed methodology. So the first is the introduction. Lung cancer is classified into two major types, the small cell lung cancer (SCLC) and the non-small cell lung cancer (NSCLC). NSCLC accounted for 85% of the lung cancer cases worldwide. It is a slow-growing cancer, which means that it is less aggressive and it is typically diagnosed at a later stage and forms a cell in the tissues of the lung. NSCLC can be classified into three major histological subtypes. The first is the squamous cell carcinoma, which accounts for 25% of the cases. It is derived from squamous cells in the main airway like the bronchi. Second is the large cell carcinoma, which accounts for 5 to 10% of the cases. The cancerous cells appears larger when viewed under a microscope. Third is the adenocarcinoma, which accounts for 40% of the cases. It is derived from the mucus-producing glandular cells. This is the epidemiology of the lung cancer. Lung cancer is the leading cause of cancer-related deaths worldwide. In Indonesia, the incidence rate and the mortality rate is 19.4 and 10.9 respectively in 100,000 people.
Okay, so now we move on to the treatment. The first one is surgery. Surgery is commonly done to remove the tumor mass. For example, wedge resection, lobectomy, and pneumonectomy. Then the second one is radiotherapy. Radiation therapy is also done but has poor survival rate and low local control. There are two types of radiotherapy, which are external radiation and internal radiations, which help to increase the local control. Then next one is chemotherapy. It is a systematic treatment injected into the patient. However, there are risks associated with chemotherapy, such as the unknown adverse effects and reduced quality of life. And finally, the immunotherapy. The immunotherapy is a specific treatment that helps your immune system to target and kill the cancer. Moreover, cancer vaccines is reported to decrease cytotoxicity. Among the vaccines available, peptide-based vaccines are gaining more interest lately due to its ability to induce broader immune response, can cover broader population, and reduce adverse reaction. Lately, immunoinformatics approach have been widely used to ease the development of multi-epitope vaccines since it is more efficient and cost-effective. It helps in reducing the time, money, and effort when compared to the conventional methods. Moreover, the in silico approaches with wide range of bioinformatics tools may help in many stages of vaccine development. It is starting from identification of the vaccine, epitopes, then the vaccine design, prediction, and the analysis. Hence, this study aims to utilize immunoinformatics methods in designing a multi-epitope peptide cancer vaccines against NSCLC for the Indonesian population.
Okay, and now we are going to discuss about the methodology. This is the overview of our methodology along with the tools that we plan to use. There are 12 main steps, and shortly we are going to discuss each step one by one in detail. Okay, first of all, we need to identify specific or unique antigen that we are targeting. Through literature review, we identify protein BPIFA1, which is a lung-specific X protein that is primarily overexpressed in NSCLC, while remain normal in other tissue, which is therefore perfect as our target. The amino acid sequence is retrieved through UniProt with accession number of Q9NP55. Moving on, since we are specifically designing vaccines for Indonesia population, HLA alleles that is predominant in Indonesia first need to be identified. We can achieve this by using the allelefrequencies.net tools, and by adjusting the country to Indonesia, we select HLA allele class 1, both A and B, as well as HLA class 2, only the DRB1 with frequencies more than 5%. After obtaining the HLA, we can now predict the epitopes which can bind to the cytotoxic T cell. For MHC class 1, the tools that we are using is NetCTLPAN. The steps are shown on the slides. Important things to be noted is the peptide length is set to 9 mer, and the threshold for selection is set to be 1%. After identifying epitopes with percent rank of 1%, the epitopes are checked for their immunogenicity using IEDB. Epitopes with positive immunogenic score is preferred to be included in the vaccine construct. Similarly, for HLA class 2, the tools that is used is the NetMHC pen. You can see the detailed steps here, and the important parameters are peptide length were set to be 15 mer. The strong and weak binder threshold are set to be 10 and 2% respectively. The epitopes that are strong binders were chosen and checked for their interferon gamma score using motif and SVM hybrid method. Epitopes with positive SVM score and Mercy score of more than 1 are preferred.
Now, I'm going to explain about the rest of the methods. So, aside from the prediction of the epitopes of MHC class 1 and 2, we also conduct the antibody binding prediction, where we predict the B-cell epitopes of the antigen. So, for this process, we're using the IEDB web server, specifically the antibody epitope prediction. And later, we will enter the antigen sequences, in this case, which is the BPIFA1 sequences in the text box. And actually, this web server provides various prediction methods. But for this study, we're choosing the bPypert linear epitope prediction 2.0 as the prediction methods, and the search will be run. And then after the results were generated, we actually select the results based on several parameters, such as the threshold must be above 0.5, and also the length of the peptide must be in the optimal length. So sometimes in the results, the B cell epitope will have a very long peptide chain or a very short peptide chain, but for this study, we're actually going to choose the optimal peptide length, where there is the peptide chain is not too long or not too short. And after the generation of the epitopes and the selection of the epitopes, we're conducting the population coverage analysis to evaluate the selected epitope coverage of Indonesian population. For this process, we're using the IEDB web server, specifically the population coverage tools. And for several parameters, we change the parameters, including the country. Since we're going to analyze the coverage of Indonesian population, the country selected will be Indonesia. And for the calculation selection, the population coverage analysis will be combining the class 1 MHC molecules and class 2. And after that, the chosen epitopes and corresponding alleles will be inserted into the text box, and the search will be run. For the expected results, actually, we're hoping that the selected epitopes can cover the population for above 90%, because since we would like to target the vaccine construct for Indonesian population, therefore we would like the population coverage to be above 90%. And after conducting the population coverage analysis, now we're going to design the vaccine construct. And in this image, we can see the general visualization of the vaccine construct design. However, this is not an accurate representation on the number of the epitopes, but this is just the general visualization of the location for the adjuvants and the linkers. So for the adjuvants of this vaccine construct will be the GM-CSF. It is found that GM-CSF is able to elicit or trigger the antibody-mediated and cell-mediated immunity. Aside from that, GM-CSF also able to enhance the initial vaccine physicochemical properties. And we're going to use 3 linkers for this vaccine construct. To link the adjuvant to the vaccine construct, we're using the EAAK linker, while to link the B cell and HTL epitopes, we're using the GPGPG linkers. And last but not least, the CTL epitopes were linked using the AAY linker. And to analyze the vaccine construct, we're using several tools, and we analyze the antigenicity, allergenicity, and physicochemical properties of the vaccine construct. For the antigenicity, we're hoping that our vaccine construct is able to elicit the immune response. So we analyze it through the Vaccine version 2.0 tools, and for several parameters we change it, such as the target organism became tumor, and for the threshold it is 0.5, because it is found that in the literature review 0.5 is the optimal threshold for this tool, because it is suitable and able to predict accurately the antigenicity of the vaccine construct. And for allergenicity, we're hoping that our vaccine construct is not able to trigger the allergic immune response, and we analyze it through the AlertTop version 2.0 tools. And last but not least, we also evaluate the physical chemical properties of the vaccine construct using the ProtParam XPC tools. So we analyze the half-life both in vivo and in vitro of the vaccine construct. We also analyze the PI, molecular weight, gravity, and the stability of the protein.
So next, the model of the vaccine construct will be predicted using PepFold server. And first, the vaccine sequence will be inputted into the system, and the advanced parameters will not be changed for the purpose of this experiment. Then the web server will predict the peptide structure through de novo peptide structure prediction. And the predicted structure of the vaccine will further be used for molecular docking with the MHC molecule. And the next step is the MHC molecule homology modeling. In this experiment, Swiss model server will be used to obtain the structure of MHC molecules. And during this step, the selected MHC sequence will be inputted to the server and run to obtain the structure that will further be used for molecular docking. The next step is molecular docking and visualization of the model. So the molecular docking between the vaccine and MHC molecule will be done using class Pro server. So first, the PDB files of the vaccine and MHC molecule structure that have been obtained previously will be uploaded to the server. And prior to docking, the desired chain will be selected if there are more than 2 chains present. And overall, the objective of molecular docking is to see the best site of interaction for docking between the vaccine and MHC molecules. Therefore, we will choose the best docking model between the vaccine and the MHC molecule by choosing the model that has the highest score generated by Class Pro. And during this step, some adjustment can also be made to obtain the best docking model for the vaccine. And the docking result model can then be visualized using PyMol. And the additional confirmation step to ensure correct protein confirmation will also be done using Ramachandran plot. So here are our references. Thank you.